For this introduction today, I'm obviously not wearing my mask. I normally do, but as you can see, I am not close to anybody, and we are certainly following social distancing and appropriate COVID-associated precautions during this unprecedented time. Thank you for joining us today to learn more about Merkel cell carcinoma and how discoveries are made about this and other cancers in a laboratory setting where we're taking information from patients and tumor samples and blood samples and putting all these things together to find the best ways to help patients deal with this cancer going forward. We have a large team of people at all levels of training, from undergraduate through faculty members and research scientists and physicians and nurses, who all come together to put this operation to work to understand this cancer better and improve treatments going forward. We welcome you on this virtual tour today of the backside of how things are happening in science. Thank you for joining.
Hello, my name is Krista LaChance, and I'm the research manager in the NEOM lab. Today I'm going to describe the patient database we use to track Merkel cell carcinoma patient information. This database is one of the most detailed in the world, for it contains over 1,500 patients with 200 unique variables per patient. And I wanted to explain what kind of information we're capturing in the database. So I wanted to give an example of a blank patient form to show you how we collect data. So we collect the patient's contact information, past medical history, so we want to know if they've had previous skin cancers or if they're immune suppressed. It's also interesting to know the patient's gender, their race, and tumor features. So the size of their tumor when they were diagnosed. We want to know if the tumor spread to the lymph nodes. And we also want to know pathological features of that tumor. We also are interested to capture the types of treatments that the patients received to their primary tumor. So whether they've received radiation therapy, surgery, or chemotherapy. And it is also important for us to know whether the patient's cancers come back. So we want to know when they had that recurrence and what kinds of treatments they've received for that recurrent disease, such as immunotherapy. And lastly, I want to point out is we capture lab results. So many of you receive the serology test, and we capture those results in the database so that we can look over time to see how the serology titer changes. So maintaining this database is a huge effort. It takes our team hours weekly to input clinic updates to the database and then also extract that data and make sure that it is accurate. One of the major efforts that our undergraduate research team takes is that they contact all patients that haven't been heard from in the past 6 months and see if there's any updates to the types of treatment they're receiving or if they've had recurrent disease. We also want to know too if the patients are doing okay. That's all good information for us. Separately, we have a blood database that is linked to this patient database, and that allows us to quickly identify interesting blood samples used for experiments. And Thomas is going to explain some of those experiments in the tissue culture station. So overall, the database is a really important tool for us to ask interesting research questions that can ultimately change Merkel cell carcinoma management. Thank you so much for your time, and I hope you enjoy the rest of your lab tour.
Hello, my name is Shira, and I'm a researcher in the NEOM lab. Today I'm going to show our pathology microscope and talk with you about the tumor microarrays. This pathology microscope, the camera, and the computer were all made possible by the generous patient donations. We use the pathology microscope to look on MCC tumors and to learn what's going on in the cells in the tumor, to diagnose the tumor cells and to learn more about the different cells that interact with the tumors. So when a patient donates a sample of a tumor, this is the way we get it. We get it embedded in parapherine in a wax that we can cut into slides and place on a glass slide that we can look under the microscope. Especially when we get a needle biopsy cores from the patients, we can place these needle cores next to each other on one slide and get to up to 100 samples on one slide of different tumor samples. This is what we call a tumor microarray. And by looking on these 100 different samples on one slide, we can get a high throughput information and learn more about the differences between the tumors and how we can adjust treatments in the future. Here I want to show you an example of these tumor microarrays. Here is an example on how the tissue microarray looks on a slide, and we can see in this purple, and here it's a little bit bigger, each core indicates on a different tumor sample, and we use hematoxylin and iosine staining to detect the cells and look on their differences. Here we're looking on each core and on the staining that we call Ki67, that it is a proliferation marker that helps us to understand how aggressively the cells are proliferating. When we zoom in, we can look specifically on one tumor sample and learn about the different cells in this tumor. The technology today allows us also to stain with a fluorescent dyes and multiple targets in order to learn about different cells that infiltrate the tumor, especially immune cells, in order to understand what type of immunotherapies we can combine and treat patients. So here is an example for this tumor microarray that was stained with these fluorescent dyes. In blue, you see the cores that stain all the cells in this sample. And we have different colors that detect different targets. So here, for example, you can see that we have the same tumor. On the right, we see purple cells that indicate on the T cells that infiltrate the tumors. And on the left, we see orange cells that are indicating on a different innate immune cells that infiltrate and support the activation of T cells. By trying to learn this interaction between the cells, we can try and understand the differences of the tumors between patients that respond or didn't respond to patient, and try to understand what combination will serve better response in future therapies. Thank you.
Hi, I'm Thomas Pulliam. I'm an MD PhD student in Dr. Paul Niem's lab, and today I'm going to tell you about the tissue culture room, which is where we are right now. This is where we process many of the samples from tumors or blood that we get from our patients with Merkel cell carcinoma, and where we actually perform experiments on these samples later on to better understand how the tumor interacts with the immune system and how we can better use the immune system to fight off the cancer. So today I'm going to tell you about two different types of cells that we study in here. The first type of cell are tumor cells, and then the second type of cells are T cells, which are part of the immune system. So tumor cells, when they're normally growing in our body, they grow uncontrollably, and that's what causes so many problems with cancer. These cells grow in places they shouldn't, and they eat up a lot of the nutrients that our body needs. And so while that's really problematic in a person, it's actually useful in the lab, because we can use these same cells again and again and again, because they just keep on growing and growing and growing. So we actually have some of these cells right here. And you can see they're in this sort of pink liquid, which is what the cells like to eat. It has a lot of things like nutrients, like sugar and vitamins in it. And so you can also see that it's sort of opaque. And that's because there's millions and millions of tumor cells in there. So we can grow these up to have up to 1 billion cells at any given time, depending on how large the experiment is. And then once we have the cells we need, we can transfer them to other little dishes like this. So this has 6 individual wells in it, and we could put tumor cells in each one of these and then treat them with different drugs. So these cells were given to me by Karina Morningstar, who's actually doing one of these experiments today. She transferred cells into each one of these, and then she treated them with different drugs to not just find out which drug kills the cancer the best, but also which drug kills the cancer in a way that it will better activate the immune system, so that any cell that isn't killed by the drug will then be killed by the immune system. And these patients will hopefully have a better response because of that. And so the immune system leads us to the second type of cell I'm going to talk about today, which are T cells. And these are the focus of my project. And so many of you who have come to these presentations before are familiar with T cells because we talk about them every year. We really like them here. And that's because T cells are such an important part of the immune system. They're really important for things like fighting off viruses like SARS-CoV-2, but also for cancer. And one of the earliest findings of our lab was that Merkel cell carcinomas that had a lot of T cells inside of them, those patients did a lot better than patients that didn't have any T cells inside of their tumor. And so that led us to the idea that these T cells are fighting off the cancer and play a really critical role in it. So the immune system plays a critical role in cancer, and so that led us to then try out new drugs like immunotherapies. These are drugs that many of you have heard of, things like Keytruda, Opdivo, and Bavencio. And these are also the drugs that had the first 2 FDA approvals of any drugs for Merkel cell carcinoma. So we study these drugs and how these T cells can fight off the cancer. But unfortunately, about half of patients that take these immunotherapy drugs, they just don't do enough for the T cells, and the cancer still progresses anyways. And so we need to figure out why some patients have responses to these drugs and others don't. And that's the focus of my project, where I study these T cells and compare the T cells from patients that respond to immunotherapy to those that don't. And so we do that by actually just studying the cells directly from people. So once we harvest T cells, either from tumors or from blood, we store them in liquid nitrogen, which is 200 degrees Celsius below 0 degrees. So it's very, very cold. And the cells will stay very stable in there for long periods of time. And I can actually show you some of the cells in here. So here's one of the tubes of cells. You can see it's very cold from liquid nitrogen. But in that tiny little vial, there's 10 million immune cells in there that we can study. So my PhD is focusing on comparing these cells from patients that respond and don't respond to immune therapy to figure out how we can better help the patients that don't respond and what sort of new treatments we can give those patients so that they can also fight off this cancer. So thank you so much. This is the tissue culture room, and we really appreciate you all attending today.